So I'm going to talk a bit about imaging, specifically PET-CT for imaging SIV and SHIV infections. So I'm going to briefly talk about some of our past work on SIV, but then I'm going to move quickly into probe development for SHIV infections. I'm going to talk about some of the considerations for imaging HIV instead of SIV. And then I want to really just touch on two issues. One is one aspect of image analysis that I think is important, and talking about how we're assessing specificity. So for those who aren't familiar with the topic, we are performing immunopet. And so immunopet, essentially we are targeting native viral molecules such as envelope, and we're doing that with an antibody or antibody-based fragment that's labeled with an appropriate PET reporter. And so when we first started doing this work, we were working mostly in SIV. We looked at a lot of different antibodies. We ended up choosing one from Jim Hoxie's lab, 73. At the time, we didn't have a primatized version of it, and so we went with whole antibody that was pegylated. And in the Nature Methods paper that was published a few years ago, we showed that you could actually administer it really up to 3 times before we saw any immune responses, any anti-antibody responses. And in this case, we used DOTA along with copper-64 in order to label the antibody. This was, I'll admit, kind of crude, but at the same time, we wanted to see, was this a viable approach for imaging SIV? Whoops, wrong button. Okay, and so we IV administer the copper-64 labeled SIV envelope binding antibodies. These are non-neutralizing, obviously, for SIV. We now are using fragments, but we'll get into that in a second. And typically we image usually 36 to 48 hours post-delivery. Typically we use about 1 mg of probe. And so in terms of you thinking about antibody treatments, this is a very small amount, relatively small amount of antibody. We're going to talk about that a little bit in terms of sensitivity that I think needs to be addressed. And usually it's 2 to 3 millicurie of activity, which just for reference point, people usually get about 15 millicuries or so of FDG, of F18 FDG for cancer imaging. So it's not a terribly high dose. At the same time, copper-64 does have a longer half-life, so that needs to be considered. In general, though, we've done some dose responses in the past, and we haven't had any issues with that. And in general, this has been well tolerated now over, I don't even know how many animals we've imaged, a lot of animals. So we've not had any issues with the probe. So I just want to briefly talk about quantification. This is, I think, an important issue in the field. We usually identify volumes of interest within an image. And again, this is 3-dimensional imaging by PET-CT. You have to identify the volume. And then the metric that's used often in PET is the SUV, which is essentially the tissue activity, so how much activity is within that volume of interest. Usually it's normalized by the injected activity, the total activity level, and by body weight. I think body weight, though, is kind of a holdover from using metabolic probes like FDG, and I'm not so sure how useful it is, but just the same, we tend to stick with it because it's the convention in the field. And in general, we're starting to, I think, look at these conventions that are used mostly for metabolic probes and for cancer, and do they really apply here? I think that's something that we need to continually be looking at. Typically, the metrics that are used for cancer are SUV max and mean, so the max signal within usually a tumor, or the mean signal within the tumor. In general, though, what we've been finding is that we started in the same place because our paper was reviewed mostly by cancer folks, to be perfectly honest. So we used the same metrics. I don't think it's really the best metric anymore, and I think I'll show you an example of why I think using total SUV, which is the total integrated SUV signal within an organ, is really more applicable. But if we had started there, I probably would have gotten yelled at by reviewers. So we went with what they were looking for. So just to give you guys a reference point in terms of what you can see from a PET-CT, these are 3 CT images. So I just want to point out that there's a lot of detail in the CT, and this is still low-dose CT, but just the same, there is still a fair amount of detail. So you can really point out many aspects of the gut. I mean, ascending colon, transverse, I have pointed out here, in descending colon. You can find the small bowel. You can easily find axillary lymph nodes, and more than one. You can actually pick out individual nodes, both and inguinal lymph nodes too. And I just showed those examples, and you can easily find the spleen. And so this is an old image when we use whole antibody. And I will note that we do have background in the liver and the heart. The backgrounds in other tissues, though, are considerably lower, and ones that are more relevant for HIV or SIV and HIV imaging. But we'll get into how we can reduce background later on. Just to note, this is just an example of, this is a region of interest that was basically detected around a region of the descending colon. I just give you an example of what this looks like in 3D, and you can measure SUV max and mean. This is for the spleen, so you can easily do that. And this is for an inguinal lymph node. So this is sort of the process that you have to go through. We'll talk about how total gets calculated in a second. But then, for those of you familiar with this work from 2015, we looked at viremic animals, we looked at non-infected animals, and we used an isotype control also. And we were easily able to detect differences across tissues that you'd expect. There is background, just as a note, in the liver and in the kidneys, so just keep that in mind. And there are ways that we're working on to try and reduce that. This was one of our first studies just under ART. It was just 3 to 4 weeks, but we could easily see decreases in signal, and those decreases were, they compared reasonably well to PCR. Obviously, we're measuring a protein, not RNA, but it was nice to see that they followed each other. Okay. So if we're going to move on to HIV, what are some of the considerations? And there are other folks. There's a group in San Francisco, I think, that's doing people, and a group in Australia. They are using whole antibody, first of all, and they're using bNAbs, and they're using basically parts and pieces that have been used in cancer. The question, though, is, and I'm really hopeful that they're successful because I think it'll really help spur the field in terms of where we started. But I think in general there are other considerations, design considerations, that we need to start thinking about in terms of how do we optimize this. And the first thing is antibody choice. So we're starting with bNAbs 2 for HIV infections. The question is, are they really the best antibodies for us to be using? We've screened them against infected cells. Many that neutralize don't bind to infected cells. Some do, some don't. And so we're looking at that. Binders may actually be better than neutralizing, maybe better than neutralizing antibodies for imaging. It's just something that we have to consider. What about antibody architecture? Well, whole antibody certainly is available when it comes to bNAbs. You can get large quantities of them. They're being made at GMP with GMP quality. So that's a big plus. There's no question. But they circulate a long time. And for imaging, that's not really ideal. And so we're also exploring other architectures like Fab and Fab2, and we'll talk about Fab2 a little bit today. Radionuclides, there's a wide range from gallium-68, which has a 67-minute half-life, to an 18F, which is 110, to copper-64 and zirconium. Most of the work done in cancer has been done with zirconium with whole antibodies. And again, the question is that the right choice for this particular type of imaging? And I'm not so sure if that's true, but I think that this is the space that we need to work in. It'd be much better to use probably a shorter half-life, maybe a FAB with a shorter half-life radionuclide, but we're going to have to do quite a bit of work to get that into people. And at the same time, there are design considerations here regarding sensitivity. Because we do know that many of these antibodies, when they bind to envelope, that the envelope is, it looks like it's recirculating. And so we do get an accumulation over time of probe within the cell. If we limit that, because we limit the circulation lifetime of the antibody, is that going to basically limit our sensitivity? And that's something we need to examine, and we're doing that at the moment. But when we talk about sensitivity and specificity, how are we assessing that? And that's what I'm going to get on to real quickly. And so we did, at least with Clade B HIV envelope, we looked at a fair number of antibodies, and there were a lot that actually bound pretty well to infected cells. This is by flow cytometry. Clade C, the only one that we found that we were really confident in was PGT121. I'm sure there are others. We didn't look at the time, we hadn't looked at PGT145. That probably would be another good one to look at, and there are likely many, many others. I think, matter of fact, in terms of bNAbs, the number of antibodies that we could screen is quite high. The point of the matter is, though, this is with, I wanted to show one example of how image analysis can impact your results. So this is, and again, this is just 4 animals, and this is just an example. I just want to use this as an example. The point of the matter is, this is acute infection, 7 days and 14 days post-infection in adult animals. This is 4 animals' viral loads at day 7 and at day 14. And I just want to point out a couple of these animals. So RIB15, there's not much of a change in its viral load over that week, but you can see more than an order of magnitude increase in, say, RYI15. So there's variation. So the question is, when we're imaging these individual animals, can we actually detect where that variation might be coming from? Can we build a hypothesis in terms of where variation's coming? If we use SUV max, you can get small changes, and it certainly looks like you're seeing changes in the spleen and in the gut that are large, and that's good to see. The question is, are those really the most important ones? And this was one animal, this is RLK15, that had about a 40x increase in its viral load, and there is an increase in the spleen. The question, though, is that these are pretty small changes when we're looking at the max. The point is are we confident that that's going to tell us what's happening or not? And I think the answer is, this is not the best way to do it. It's not wrong, but it's probably not the best way to analyze the data. And so we moved to total SUV, and the only thing I'm going to say about it is that it's a pain in the butt to do. There's a fair amount of work that goes into this, and we are using a number of different algorithms, especially for the gut and the NALT, and a few other areas that take a little bit of interpretation. So I would say this isn't quite automated yet for the gut, but there is we're working on that. We're trying to make it as automated as possible. The lymph nodes and spleen are pretty easy because they can be easily guided by CT, but it's more difficult in the gut. If we look at now using total SUV, first of all, I want to note that the total signal in the spleen now is not the dominant one. So there's a lot of signal in the spleen. I don't want to get that. I don't want to make change your minds on that. And there's a large increase. But the point of the matter is that when you integrate all that signal in the gut, now you start to see that the gut is really dominant in terms of the amount of total virus that's there, or at least total envelope, I should say. We're imaging envelope. And so I think this gives you a better, more accurate perspective of the data. That's really what I want, really the point I want to make. And when we compared the 40x increase in viral load to the 1.65 increase viral load animals, now I understand these are individual animals. I don't want to read into it too much, but I just want you to see when we're following a single animal, I think there are some insights, and again, it's hypothesis generating. What you see is a huge increase in the spleen in this animal. I mean, it really skyrockets. So if we want to start to answer questions about how virus gets from tissues into the bloodstream and how that happens and where that's coming from, I think this can be a useful tool. In the gut, there weren't huge increases actually over that time span. So, and can you see this in the images? And yes, so this is signal in the gut from these two animals. This is the change in the spleen that increased quite a lot. And you can see even this axillary lymph node actually increases quite a bit too. But again, this has to be evaluated in 3D, and it has to be integrated over the whole organ. We did see actually changes in the lung in this particular animal. And as a matter of fact, we see signal in the bronchial lymph nodes, which I don't think gets looked at terribly often, but we can clearly see it. I don't know how much time I have-ish. 3 minutes? Oh, that's a shame. Anyway, I have so much more to talk about. So let me just skip a bit. So the point is, you can these differences among animals are actually pretty easy. The viral loads in this animal had dropped quite a lot, and so you can see changes that are really easily detectable. This is using PGT121. This is another animal whose viral load really was undetectable at this point. There's not much signal in it. This was on day 16. I can talk more about it. We are comparing across antibodies. This is PGT121, 3B and C117, VRC01. The viral loads are down here. The viral loads are pretty low, and we can still pick up. This is 115 using VRC01, and we do find pockets. This is in the gut of signal. Some of the puncta here are actually mesenteric lymph nodes. And we are comparing the data across these antibodies and looking at various organs. And we are going to have to try and figure out how we're going to connect this to viral load. So it's not trivial. In the last few minutes, I just want to talk about specificity. So what we're doing, and I think this is important, is we're trying to connect PET to flow cytometry. You guys know what flow cytometry is. You're used to using flow cytometry. You're not used to looking at PET images. So how can we connect the two? We combine PET-labeled probes along with near-infrared fluor-labeled probes. We combine the two. And you can have, here's a monkey that we image with PET. We can take the organs out, and we can move this through a flow panel and look at how we can confirm whether or not those cells are envelope positive. And the way we're doing that is that we verify the specificity using an ex vivo probe, usually against a different epitope, and make sure that it binds. So we're comparing the in vivo probe with the ex vivo probe. We're also going to be adding GAG staining too. You can do even more if you want, but we can add this into sequencing also. But the whole idea is to look at these positive predictive values. I just want to give you one example from SIV. This is using a primatized version of 73, so a new version of that antibody as a Fab2. And then we used 411 as our ex vivo probe, essentially. This was a fairly reasonable viral load. the point of the matter is that this is the ex vivo positive cells, this is the in vivo positive, and these are the positive predictive values. So we get about 96% specificity. Our sensitivity was about 76%, and what I mean by that is that it actually is underestimating. So we're underestimating the number of positive cells, which I'll be honest, I'll take that as opposed to overestimating. And so only hitting 76% may mean that we need to look at dose, and the percent that were CD4 positive was about 98%. So the point is that we can use this to look at basically cells from individual hot tissues and get these types of numbers. And I think this is important in terms of really nailing down our specificity and sensitivity. And so we're going to be able to look at dose, and I can just say we're doing this with the other probes too. And so with PGT-121 at 23,000 viral load, we saw about 95%, this is just specificity. We went down to 290 and it dropped to 88%. And so that's something that we need to look at in terms of as viral load drops, how does our specificity change? What might be affecting that too? So that's, I think, important for us to look at. But the point is we can quantify this. So we'll be able to give you numbers. And I think that's what folks, in terms of specificity and sensitivity, people have complained about. We need numbers. Looking at images is nice, and even though we quantify them, it's kind of fuzzy to folks. So I think this kind of information will be helpful in showing you how specific and how sensitive we are. And it'll help us in terms of comparing cross probes and comparing across probes and viral loads too, and across viruses. So I will stop there and thank folks in my lab and the Villager lab for this work. Thank you, and NIH for funding it.
Time maybe for one question.
I didn't go over too badly.
Keep pushing.
Yeah, exactly.
So what do we have to do to collaborate with you?
Just talk to me and Francois and anybody. So yeah, it's fine. Just talk to us, I think. I mean, we're collaborating with Tom. Tom's going to bludgeon you with more imaging in just a second. Oh, okay. So I think we're pretty collaborative. So yeah, that shouldn't be an issue.
Well, we always need money.
Yeah, monkeys aren't free, but you guys already noted that earlier.
Which is really great. You don't have to get up at 3 in the morning to go to the local hospital to see monkeys.
Yeah, I should note the support of NIH on that. Okay.